Yeah, so I begin by thanking the organizers for this invitation and for the putting together this meeting, despite, as I understand, very tough circumstances. Yeah, so I'll be talking about myosin II independent cyclic contractility of actin filaments in membrane nanotubes. So I begin by explaining what are membrane nanotubes and how we generate them. So the technique is very straightforward. What you do is you have a cell here. You come with a plastic bead, which is about a micron in diameter, and you trap it in an optical trap. And then you come with the bead, you stick it to the membrane, and then you change the distance between the cell and the bead. So in this case, the cell is moved away from the trap bead. And when you do that, what happens is you extract a membrane tube out of the cell. So this can be done in very different ways. Here I have showed the use of optical tweezers. You can also do this with glass microneedle. And here is a motorized stage. There is a fine glass needle. And you can stick the tip of the needle to the membrane and you can pull and you can extract a nanotube. Typically, these tubes have a diameter of about 100 nanometer, and the length depends on how much you pull. And these different techniques are useful because here in this case we use a confocal microscope, and the tweezer is not built on the confocal microscope. That is why we use different ways of pulling. So this is a schematic like what we use neuronal cells, and so that is the axon of a neuronal cell. And what you do is, again, like I mentioned, you stick the bead and you pull, and this is a tether which you pull out. So normally when you do these experiments, what you see is that when you pull, there is a sudden sharp increase in force as the tube is drawn out of the cell, and then this force will settle down, and then you measure a steady state force thereafter. And you can apply different amounts of pull. You can first pull a tube, then you pull it again and you pull it again. You see that every time the force comes back to a baseline value, suggesting that the cell maintains some sort of a tension homeostasis. Here is a video of a tether being extracted, and you can see that this is the thin tube which is pulled out. So in the experiments which I'm going to describe, what we do is we pull the tube like this, and we keep the distance between the axon and the trap constant. Okay. So you extract a tether and you don't do anything more. You just wait and you keep measuring force. Okay. That's the experiment. So to our surprise, what we saw was when we did these experiments on these neuronal tethers, the force did not remain constant, but instead the force showed these kind of repeated load and fail behavior. Okay. The force increases, reaches some value, suddenly collapses, and then again increases, and this kind of cycle keeps repeating. I mentioned earlier that in most cases, this does not happen. If the displacement is kept constant, then the force is constant. So this came as a surprise. So you can, when you see this kind of active force generation, the first thing to do is to go in with various drugs and see what is important and what is not important. So the first plot is the control. And then we used the drug called blebistatin, which inhibits myosin II molecular motors, because what you're seeing is some contractile forces. So we thought maybe molecular motors are involved. And you see that this drug does not really prevent this kind of force generation. You can inhibit myosin by different ways. So we tried different mechanisms of inhibition of myosin II. We also tried, there are only few drugs available for unconventional myosins. In the case of myosin 5, there is a drug available, so we tried that, and that also didn't do anything. We tried inhibiting nucleators of actin, and that also doesn't seem to do, at least it doesn't prevent these active force generation. What really kills the active force generation are treatment with latrinulin A, which depolymerizes actin filaments, or treatment with this drug called jasplakinolide, which stabilizes actin filaments. It doesn't allow actin filaments to undergo turnover. So to summarize this, that we don't observe any load and fail behavior only if we either deplete ATP, we depolymerize actin filaments, or we stabilize actin filaments. But myosin II has no effect. So then we went on to image actin inside these filaments because actin seems to be important. And this is what we see. This is actin, which you're seeing as bright. And you see that actin suddenly retracts from the tip of the, this is the tip of the tether. And you see these cycles. Actin grows inside the tether, reaches all the way to the tip, and it suddenly retracts back. And these cycles keep repeating. So you can now make a sequence of images, and here you can see that these cycles are repeating. Actin is growing. This is the base, the axon, and that is the tip of the tether. And you see actin grows, it retracts, then again grows again, and so on. And this is more clearly seen in this inverted kymograph. And you can see that the retraction velocities are high at the tip of the tether, and the velocity is almost zero as you go towards the base of the tether. So it's not as if something is pulling the actin from the base. It's some mechanism which is operating within the tether. So then we can try to correlate the force and this actin dynamics. So these images were made by using the glass needle method. That is why you don't see the trapped bead here. Here we used optical trap, and this is the base of the tether, the axon. And that's a tip, and the tip is looking bright because the bead which is trapped in the optical trap also scatters a lot of light. That's the reason why this is looking bright. But what I want you to focus on is what is happening inside this tube. And you see that the actin grows, and you form a bridge which spans the entire tube. And that is when the force begins to increase. And when the force collapse corresponds to the detachment of actin from the tip. Okay. So what is happening is actin is growing inside the tube, making contact with the bead. It starts pulling on the bead, and suddenly the actin detaches, and then you see that event as a force drop, and then the actin retracts back, and then the actin grows back again. So you can, this behavior is summarized here. These are intensity plots. The intensity, this is the distance from the base of the tether. 0 is the base of the tether, and that's a tip. And the time goes this way. So these are intensity plots taken at different time points. So you see that initially you had actin growing all the way to the tip, and the intensity sort of falls off, suggesting that there are many actin filaments which are growing in the tube, and the actin density decreases towards the tip of the tube. And then at some point you see these retraction events where here you can see it more clearly, that the actin retracts within the tube. So here there is hardly any intensity of actin, and actin becomes concentrated in these peak-like regions, and these peak-like regions move back. And you see that there is very little intensity ahead of this peak, suggesting that it's not depolymerization, but the filaments themselves are being pulled back. Because had it been depolymerization, you would have seen fluorescence coming from the monomers appearing here. So this suggests that actin filaments retract within the tube, and after they retract, they kind of redistribute, and then it becomes again this continuous monotonic fall from the base to the tip. So there is some process by which actin is kind of retracting, and then it seems to depolymerize and then grow back and then again retract. So you can ask the question, what happens if I pull extremely long tubes? So this is a case where the tube is, you cannot see the tip of the needle, but the tip is somewhere here. So this is about 30 micron long tube. And in this case also you see this actin growth and retraction cycle. So this growth and retraction process has nothing to do with the bead sitting at the tip of the tether. The bead just allows us to measure the force. We also see helical buckling of the tube. As the tube goes back, you can see this in this video probably. Oh, sorry. You can see that here the whole bundle buckles to form helices, and you can quantify this, and you see that this shows the distribution like this. There is some characteristic length at which these bundles undergo buckling. So now I just want to give an introduction to another tubular protrusion which is spontaneously generated by cells and make a comparison with that. So these are called philopodia, and here is a video of a growing tip of an axon, and you see these tube-like protrusions being spontaneously generated from this growth This entity is called the growth cone. So people have been studying these structures for a very long time in all kinds of cell types. And if you look inside these structures, what you see is something like this. You have actin filaments, and these actin filaments are polymerizing from the connected to the base of the filopodia, and they're polymerizing from the tip, driven by polymerizing factors like formins, for example. And so you have a bundle of actin filaments, and the polymerization of these actin filaments generates a force, a pushing force on the membrane, and it is this pushing force which generates these tubes which you saw in the previous video, the extension of the tubes. And there are a number of other components inside. There are cross-linking proteins which cross-link these filaments, and there are unconventional myosins which can bind to the membrane, and they can walk on these filaments. And in here you see that these punctures at the tip, which is caused because these myosins, myosin 10, for example, transport certain proteins to the tip of the filopodia. So the growth mechanism of these sort of tubes are fairly well understood. But what is interesting is that these tubes also grow and retract. And while they retract, they can generate forces on anything which is stuck to the tip of this. And this retraction mechanism and the pulling force generated by these tubes is much less understood. So here are some early experiments. So in this experiment, what they did is they put a cell which is producing these filopodia, and this cell is sticking on a deformable substrate. And this deformable substrates have some fluorescent beads embedded in it. So you can, whenever a filopodia is pulling, the bead which is embedded in the substrate gets displaced, and you can measure this displacement. And here you see these kind of sawtooth-like pulling events which is happening. And the explanation given in this paper is that there are myosin motors sitting at the base of the filopodia. You can't really see the filopodia here. You should imagine that there are thin tubes connecting these red dots. There are myosins sitting at the base of the filopodia, and these myosins are pulling. In this paper, they never tried blocking the myosin motors, so experimentally they don't have any evidence for it. But as I mentioned, in our case, putting blebistatin and blocking these myosin 2 motors did not have any effect. So this kind of mechanism does not explain the pulling which we see. There is another mechanism which has been proposed by the group of Patricia Becerril, where they say that there is retrograde flow happening at the base of the tube. That what I mean is here, this is connected to a lamellar podium, and there is R23 mediated polymerization happening from these membranes. So there is a retrograde flow happening here, and these filaments which are inside the tube gets coupled to this retrograde flow. So these filaments are getting pulled at the base, and so they suggest that there is a constant pulling at the base, and this is what causes the pulling forces which you see in filopodia. This again does not fit with our experiments, because if something is pulling at the base, then you would have seen the maximum velocity at the base of the tube. But as you saw from the kymograph, the velocity is nearly 0 at the base, and the velocity is maximum at the tip. So anything myosin or retrograde flow pulling the filaments at the base of the tether, it doesn't fit with the experimental observation. Then there are other mechanisms proposed. Here they invoke activity of chiral filaments resulting in stresses which generates torsion within the tube. And here they propose a mechanism where you have these cross-linking proteins which connects the tip of the actin filament to the membrane. And as the actin undergoes depolymerization, this generates a pulling force on the membrane, because these linkers remain connected to the actin filaments as the actin filament depolymerizes. This is somewhat analogous to the pulling of kinetochores by microtubules, depolymerizing microtubules. But this again does not fit with our experiments, because we see that even in long tubes where the actin cannot reach the tip of the tether, we see this growth and retraction cycle. So this growth and retraction cycles does not depend on whether the tip is connected to something or not. So here I summarize these things, that blebistatin or blocking myosin motors has no effect. So it's myosin 2 independent. F-actin bundle buckles and actin retracts. The velocity is minimum at the base. So this rules out pulling at the base. There is a delay between these load and fail events. So any mechanism which we propose has to explain why these cycles happen, why it is cyclic and not just a one-time pulling of the filaments. You can look at, you can consider these myosin, unconventional myosin motors, which are bound to the membrane and working on actin, but you can estimate the force these motors can generate because they are stuck to a fluid membrane. So when they walk, they just simply advect. They cannot generate forces because the cargo domain is attached to a fluid sheet. And these forces, which when we estimate, it comes out to be extremely small. I already mentioned that the tip-based mechanisms are unlikely because even very long tubes show this growth and cyclic behavior. So we have to look for other possible explanation. So then we realized that there are other mechanisms of stress generation within actin filaments. It is very well known that there is this class of proteins called ADF cofilin, and what they do is they bind to actin filaments. And when they bind to actin filaments, they bind to this pocket within each actin monomer, and by binding, they sort of create some sort of allosteric deformation of the monomer. And this deformation created in each monomer sort of manifests as itself as a change in the helical pitch of the actin filament. And when cofilin decorates the filaments, there is a change in pitch, which is as much as, I think, something around 20% or so. And this has been seen in electron micrographs. It has also been seen in fast AFM studies. So what they suggest is that when these proteins bind to the filament, it generates a torsional stress on the actin filament. This changes the helical pitch of the actin filament. And when the torsional stress is large enough, the actin filament disintegrates. And one of the jobs of cofilin inside a cell is to regulate the dynamics of actin filaments by severing actin filaments. And the way it severs is through this kind of a mechanism. It's also important to note that cofilin or ADF, they're similar proteins. These proteins cannot bind to a freshly polymerized actin. This is because a freshly polymerized actin is ATP actin. And as time progresses, ATP gets hydrolyzed. Then you get the ADP plus pi F actin, and then you eventually get ADP actin. And cofilin binds only to ADP actin. This means that this mechanism of cofilin binding, it involves a delay because the freshly polymerized actin filaments are immune to severing by cofilin, and only after hydrolysis has happened, cofilin can bind and start generating stresses within the filament. There has been some more recent experiments which was done on actin filaments, which is held between an optical trap and some stuck bead here. So this is a filament, and what they did was to apply different amount of tension on this filament, and then pass cofilin into the chamber and see how cofilin binds and severs a filament. And what they see is that if the filament is not held taut, if the filament is slack, then cofilin is very efficient in severing these filaments. And the moment you apply a tension on the actin filament, the efficiency of severing goes down. And this suggests that there is some sort of coupling between the longitudinal tension which is experienced by the filament and the ability or the probability by which cofilin can sever the filament or bind to the filament. So based on all these things, we came up with the following hypothesis, that what is happening is F-actin is the filamentous actin polymerizes from the base of the tube. As the polymerization proceeds, ATP actin gets converted to ADP actin. Then cofilin or ADF starts binding to this ADP actin and starts over twisting the filament and generating stress in the filament. And it is this stress which we see as a pulling force on the bead. As the torsional stress increases, the F-actin detaches and fragments, because now the stress is very high, and either the filament detaches from the bead or the filament just fragments because of the action of the action of cofilin. And that, as I mentioned, that is what cofilin is the purpose of cofilin inside the cells. And F-actin will degenerate and it starts polymerizing fresh from the base, and the cycle repeats. So this kind of a hypothesis can explain why you have the cyclic load and fail behavior. So we imaged ADF inside these cells, and we see that, so this is, I think red is actin and green is ADF. But what you see here is that as actin polymerizes inside the tube, you also see ADF going inside. and these two are correlated. The intensity fluctuation of actin is correlated with the intensity fluctuations in ADF. Then we used inactive forms of ADF, and when we use an inactive form of ADF, we see that the force generation decreases. There's much less force generated. Remember that the inactive form is competing with the wild type or the natural form. So it's only a competitive inhibition. So you see that the peak force decreases when you express this inactive form, and you also see that the peak duration increases. That is, now the force is generated and the force remains high for very long time, like as high as like in this case, I don't know, 100, or we have seen up to 200 seconds of force events. And this is not the case in untreated cells. In untreated cells, the force increases and very quickly it falls off. So it suggests that in this case, the filaments are not getting severed efficiently, and because it's not getting severed and it's bound to ADF, the force can be sustained for longer duration. You can also express a constitutively active form, and this does nothing because, and that is not too surprising, because if there is enough ADF there already to decorate the filaments, then expressing more active form of ADF is not going to make any difference. So based on this, we came up with this kind of a simple-minded model. You have an actin filament, and when the actin filament grows from the base and spans the entire tube, so the green part is the membrane tether, and as the filament grows and ATP gets hydrolyzed, cofilin starts binding to these filaments, and what cofilin does is that it reduces the length of each monomer. So each monomer shrinks by some amount delta L. Okay. And cofilin can bind and unbind with some characteristic time constants, and this filament is attached to at the tip via some linking proteins, and eventually it's attached to the trap. And we also assume that this linking protein, which links the actin filament to the tip of the tether, it also has an on-off dynamics. So you can put it in forms of equation that for a single filament, you can say that the fraction of this epsilon is the fraction of cofilin bound monomers, and delta L is the amount which each monomer shrinks, and L is the size of each monomer. And with this, you can write this kind of the equation for the time evolution of this cofilin binding, and you can calculate that because of this shrinkage of each monomer, how much force is generated at the tip of the filament. Okay. This is for a single filament. For multiple filaments, the things get a little bit more complicated because the load is shared by different filaments, and these filaments are not binding exactly at the same time. But I'm not showing the full model, but that can be taken care of in a numerical simulation. And when you do that, what we see is that we get exactly the kind of force responses which we see in experiments. You see these kind of saturation events where the force increases and tend to saturate and then collapses. You see this sort of sudden changes in slope, which you see in the model also. The right side are the plots generated by the model, and left side are the experiment. And you see this sort of kind of partial drops and regrowth. And these can be understood because this, for example, is a case where one of the filaments detaches. So the rate of generation of force decreases when a filament detaches. Here another filament gets attached and then the rate increases and so on. And these are cases where there are if a filament detaches and the load is now shared by the filaments which are remaining. I will not go into details of this because you can also fit this to the distributions which we see in the experiments, and it seems to kind of agree with that. So yeah, I'm running out of time, so I'll summarize. So active force generation due to small number of force generating elements explains the kind of the time series which we see, force versus time plot. Mechanisms that pull at the base are unlikely. Stresses generated in the actin filaments by ADF cofilin is a possible mechanism. And the best way to test this further is to use in vitro experiments. So we have just built a double trap optical tweezers where you can trap two beads. You can hold an actin filament in between, and you can flow in cofilin, and the hope is that we will be able to check whether there is any stresses generated in the filament by doing this experiment. So this is something which is just, the setup is there and we plan to do this, and if any interested students want to join, they're most welcome. And with that, I thank all the collaborators, Most of the experiments were done by these two people. Serene is the one who helped with all these ADF constructs with genetic interference and so on. The model was developed by Pearsons at Institute Curie and his postdoc Tapas Singha, who's also at Institute Curie. We thank James Bamberg for giving us all these various ADF constructs and Jacques Pro for several discussions. Thank you.
We have time.
Really nice. Did you say what cells you're using?
The cells we are using are chick DRG neurons, dorsal root ganglia neurons, taken from chick embryo.
Okay.
So these are primary cells.
Okay, nice. And I'm wondering, is ADP release force dependent?
So I showed you that the paper from the Japanese group where they hold an actin filament and you pull on it, and there they see that there is whether the ADF, actin depolymerizing factor, whether it can bind to the actin filament or not depends on the force. And this is interesting because this also is another way by which you can have mechanosensation, because in cells, if you take a fibroblast and if you label for ADF, then we see that there's a lot of ADF in the filopodia and the lamellipodia. These are the dynamic parts of the cell, but you never see ADF decorating the stress fibers. Okay. And there is an old paper which suggests that ADF doesn't decorate stress fibers because stress fibers are under tension and therefore ADF doesn't bind. And this probably is important for the cell because otherwise the stress fiber lifetime will be very small because the filaments are getting severed all the time.
Good way of distinguishing.
Yeah, so I think, so we believe that there is this coupling between tension in the filament and ADF's ability to bind to the filament.
Pramod, the model that you've written down at least doesn't seem to have any active features. It's,
the active feature comes indirectly because of the fact that you allow for this dynamics to repeat. So the force generation,
right.
The force generation comes from the binding of the protein.
Right.
So you can think of that as just some competition between the binding energy and some elastic energy.
Correct.
Right.
Yeah.
But in the cell, for this cycle to repeat, you need some activity.
No, in the cell of fine.
So it's not explicitly there in the model.
It doesn't seem to be in there in the model.
That's correct.
Right. So which is why it might be interesting to look at have an experiment where you see the differences between rather than exact comparison between the model and the,
right.
As you're saying, the cell there might be something more interesting going on.
Right.
So for instance, if you measure the fluctuation response ratio, which is an indicator of how active the system is.
Right.
Because I don't think the model that you have will give you a sort of active, if you measure the fluctuation response ratio, in equilibrium it goes as 1 over the temperature. But in an active system...
Yeah, so this model may not give that. I agree. Because as I said, the activity is not explicitly there.
Yeah, exactly.
Yeah. But the real system has activity because ATP hydrolysis is happening.
Exactly. So it might be good to do that hydrolysis.
I agree with you.
I was intrigued by the observation that myosin II doesn't seem to play a role because in vivo there's quite strong evidence for that in different cell types like the heart and so on. So okay, is that better? There we go. So in vivo, there's quite strong evidence for myosin II playing a role, mutants and all sorts of things. So do you think this is something that's specific to the type of cell you're looking at? Or do you think there's actually a general mechanism here that in vivo has kind of been missed?
We do not know whether it is specific to these cells which we are looking at or not. But this sort of load and fail behavior has been seen in some other systems also, like I mentioned filopodia. And in the case of filopodia, for example, the experiments I showed from Patricia Bassaro's group, there they clearly see there is a retrograde flow at the base of the tube. So I do not know at this stage whether there are 2 or more mechanisms which can operate simultaneously.
Because in vivo there's quite a lot of evidence where if you see myosin II localized at the base of the filopodia retracts.
Right.
Now, you could argue that's correlation, but it's pretty strong correlation.
Right.
And so I guess the question is, do you think it's a different mechanism or do you think it's somehow complementary?
Can you please repeat the last part?
Do you think that different cells have different mechanisms of regulating filopodia, or do you think that there may be some complementary process that's been missed?
So one big difference between axons and these other cells is that in these other cells, the filopodia is coming out of a lamellipodium. Okay. Whereas in the case of axon, the axonal shaft is very different from a lamellipodium. There's very little actin at there's a very fine actin cortex, which mostly in the form of actin rings, and then it's mostly microtubules. So mechanisms like retrograde flow and these sort of mechanisms may not apply to the case of axon. So there could be differences arising out of, differences from cell to cell.
One quick question. So in filopodia, that search or don't search. So the people, I think, have seen differences in the association of cofilin with actin. The point that they call it cofilactin or something. Mm-hmm. So I was just wondering whether you see differences in distribution of cofilin along.
We don't know. I showed you the images where we have tried to label both cofilin and actin. And what we see is that if you look at now there are many filaments which are polymerizing and depolymerizing, and they're not all in registry. But what we do is we draw a small box and we look at the intensity as a function of time for each of these components. And what we see is that these two intensities are pretty much correlated. We were expecting to see a lag between these two processes because the mechanism which I proposed, where the actin polymerizes, it's ATP actin, and cofilin should take some time before it binds. We don't see a very clear lag. There seems to be some lag when the intensity is going up, but it's not very, it's not something which we can say with confidence. So this is something which is, probably it needs more experiments to look at, it has to be looked at more carefully whether this signature is there actually or not. What is the association between cofilin and actin in these nanotubes, right? As a function of time.
Okay, there are no more questions. Let's thank Pramod for a fantastic talk.